Hello everyone, I am Shubham and I welcome you all to another Research Spotlight episode of Synthesis Workshop. Today we are joined by Dr. Roman Sarott and Dr. Sai Gourisankar, former and current postdoctoral associates in the Gray Laboratory at Stanford University. Dr. Roman Sarott is currently the research group leader at Max Planck Institute for Medical Research. Today, they are going to present a talk on relocalizing transcriptional kinases to activate apoptosis. With this brief introduction, I hand over the stage to Roman and Sai to begin their talk.
Thank you for hosting. We're delighted to present our recent work, Relocalizing Transcriptional Kinases to Activate Apoptosis. Roman and I co-led this work, and we'll be switching off during this episode. Stories about cancer. More than 40 years of molecular genetics has identified drivers of cancer progression, genes that when mutated cause proliferation, survival, invasion, and metastasis of a tumor. And there are many categories of these hallmarks of cancer. These genes could be involved in cell motility or cell differentiation or viability. But one commonality that has emerged is that they converge on changes in gene expression. So we were interested in developing targeted cancer therapies, precision medicines that target gene expression downstream of cancer-driving signaling, because we felt that this would strike at the heart of a malignant cell. Now, if you go back to the literature, you'll find that most approaches currently to target gene expression try to shut down malignant transcription. These molecules can largely be categorized into two different mechanisms. The first class are inhibitors, which competitively occupy an active site of an enzyme that's important in epigenetic regulation, such as a transcriptional kinase such as CDK9, shown here in the middle in light blue. The second class are degraders, which might destroy the epigenetic protein of interest by recruiting it to the cell's E3 ligase/proteasomal machinery, and ultimately aim to phenocopy genetic knockout. And this strategy has been enormously successful, especially in the recent decade. But both mechanisms, both pharmacological strategies, result in a loss of function effect, loss of function pharmacology. And this type of pharmacology can be enormously potent, but also can be toxic, especially if the enzyme or protein being targeted is important in healthy cells, which is the case for a large number of regulators of transcription in particular. And this is because, of course, the effect of your molecule is generally correlated with how well that molecule inhibits or degrades the target protein. So we wanted to take a different approach. We knew that programmed cell death, or apoptosis, is actually a feature of all cells in the body, both healthy and cancerous. Programmed death is actually critical for the development of multicellular organisms, for human development. It allows the body to get rid of cells that have faced acute DNA damage or stress. But of course, in cancer cells, the cell death signaling pathways are often bypassed or silenced. So our idea and the hypothesis at the heart of this presentation was that we could rewire or relocalize or hijack cancer drivers to activate apoptosis, turning the biochemical signals that ordinarily might turn on the expression of proliferation or survival or invasion genes into signals that turn on apoptosis and cause the same cancer cell to commit suicide. We wanted to do this using a bivalent small molecule. The goal was to make a molecule that was two-sided in nature, one side binding a cancer-driving epigenetic activator, and the other side binding a death regulatory transcription factor that binds DNA at the promoters of a pro-apoptotic gene. The induced proximity of the two proteins, we hypothesized, the activator on the left, shown here as TFA, the det regulator on the right, would be sufficient, we hypothesized, to turn on the expression of a programmed cell death gene expression program. Now, of course, to do this, we needed a death regulatory transcription factor. And so what could that death regulator be? We looked through the tumor molecular genetic data and the literature and landed on a transcription factor called BCL6 or B cell lymphoma 6. This is a zinc finger transcription factor, TF for short, I'll use that abbreviation later in this presentation, that binds DNA at a consensus site, shown here, and epigenetically silences the transcription of death and growth arrest genes at those sites in cells. And it does this, it carries out that silencing program by virtue of an N-terminal structured domain called BTB, which has biochemical affinities for copressor complexes such as polycomb or histone deacetylases. Now, as you might guess from its name, BCL6 is deregulated in diffuse large B-cell lymphomas, which is the most common human lymphoma. And I think you could imagine how deregulation of this transcription factor that ordinarily silences death genes now causes these cancer cells to continue to proliferate when otherwise they might commit suicide. Recently, pharmaceutical and a number of academic groups have endeavored to develop inhibitors that competitively bind the N-terminal BTB domain of BCL6 to displace those co-repressor complexes I mentioned. Their hypothesis, as you might be able to imagine from this presentation, is that displacing these co-repressors would allow the expression of death genes and kill the tumor cell. These groups have developed a number of biochemically potent inhibitors of the BTB domain co-repressor interaction. One is shown here from Boehringer-Ingelheim with the IC50 of 2.9 nanomolar. But they've exhibited rather weak anti-tumor effects in cells or in vivo. Degraders recently developed, not shown on this slide, have had similar characteristics. We hypothesized that inhibiting BCL6 is not enough. We should instead flip the effect of BCL6 from a repressor to an activator using a bivalent small molecule. And so we hypothesized that we could use the inhibitor that binds to the BTB domain to synthesize a bivalent small molecule, a two-sided molecule that relocalizes a transcriptional activator to those promoters that BCL6 binds to. And so by relocalizing the activating ability of, for example, a transcriptional kinase, such as labeled on this slide, CDK9 with its partner cyclin T, we could push the gas pedal on programmed cell death gene transcription, enabling paused polymerase to elongate the gene body. The principle at stake here, again, the core of our hypothesis, is induced proximity, or in this case, chemical-induced proximity, the simple biophysical co-localization of proteins inside the cell, which a number of studies over 30 years of research has shown is sufficient for dozens of biological processes from the membrane to the nucleus, including especially epigenetic regulation and transcription. And so we set out to synthesize these bivalent CYPs, chemical inducers of proximity. We call them transcriptional chemical inducers of proximity, or TCYPs for short, to relocalize CDK9 to BCL6 bound DNA, activate RNA polymerase II, and turn on the expression of pluriapoptotic genes. Here I will hand it to Roman to describe how we engineered such molecules.
Thank you very much, Sai. Now, while figuring out how to realize our hypothesis, we were faced with a major complication, and that is that there are only ATP competitive inhibitors of CDK9 developed to shut down its transcriptional activity, the exact opposite we were trying to achieve. However, known CDK9 inhibitors differ greatly in potency, binding kinetics, and selectivity, and we took the calculated risk that through suitable inhibitor and linker choice, we could develop molecules that recruit CDK9 and its activity. And so we embarked on a medicinal chemistry campaign which generated over 100 bivalent compounds, connecting CDK9 inhibitors here in blue to BCL6 ligands in red, with linkers varying in length and chemical composition. And we then evaluated this library in an assay triad that in our hands has proven efficient for developing transcriptional inducers of proximity, looking at BCL6 dependent transcriptional activation, ternary complex formation, and cell killing. And these studies nominated a lead compound, CDK-TCIP1 here in red, which links the CDK9 inhibitor SNS-032 to the BCL6 binder BI3812 through a flexible carbon linker. And additionally, we synthesized two negative controls in which small modifications negate interactions with either BCL6 or CDK9 through minor structural modifications, shown here in red. In a BCL6 overexpressing reporter cell in which we introduced a plasmid with a gene encoding green fluorescent protein downstream of BCL6 binding sites, CDK-TCIP1 robustly activated BCL6 controlled GFP expression, whereas the negative controls do not. And this is a very cool result, as it shows that chemically induced proximity enables turning an ATP competitive kinase inhibitor, SNS-032, into a locus-specific activator of transcription. I also want to direct your attention to the characteristic bell-shaped curve, which is often referred to as a hook effect, indicating that saturating levels of the compound occupy both proteins separately. And this is a hallmark of bivalent inducer proximity, prompting us early on to propose that a transcription-competent ternary complex between CDK9 and BCL6 is necessary for the observed effect. And this feature is not unique to SNS-032, and we can turn structurally diverse CDK9 inhibitors into activators of transcription, as shown here using the same GFP reporter cell line. We then developed a cellular nanobread assay with full-length BCL6 and CDK9 and looked at the ternary complex formation of the bivalent molecules. And at this level of granularity, we found the propensity for ternary complex formation is predictive for induction of GFP and thus of transcriptional activation. Now, coming back to our lead compound CDK-TCIP1, at this point we knew it activates BCL6 dependent transcription and forms a ternary complex in cells, and also that this ternary complex is likely crucial for activity. Lastly, and maybe most importantly, CDK-TCIP1 potently kills BCL6 overexpressing human lymphoma cells at single-digit nanomolar concentrations, over 250 times more potently than the additive effect of BCL6 and CDK9 inhibition, here in black. And that is despite the large difference in cellular permeability between traditional inhibitors and bivalent molecules. So we were fairly sure to be onto something interesting. But potency is of course only half the equation in oncology, especially when targeting essential proteins such as CDK9. And to explore the specificity for DLBCL cells, we then collaborated with the PRISM consortium at the Broad Institute, where they simultaneously screened drug sensitivities in almost 1,000 barcoded cancer cell lineages. In this graph here, each column is a cancer lineage, and the longer the pink bar, the more are cell lines in a given lineage sensitive to CDK-TCIP1. And we found, as you can see here, that across all these lines, uniquely and exclusively, lymphoid cancer cells with deregulated BCL6 were sensitive to our compound. In other words, CDK-TCIP1 is not just very potent, but also surprisingly specific. We were thus intrigued to explore whether our approach is amenable to modulate BCL6-mediated processes in vivo, particularly in mice. However, the physicochemical properties of bivalent drugs, including those of degraders, are rarely optimal to begin with. And this was also the case for CDK-TCIP1, whose metabolic lability prevented in vivo use. And so, focusing on cyclized and amine-containing linker motifs, we synthesized an array of derivatives that proved to be significantly better activators of BCL6-dependent GFP expression, as well as killers of DLBCL cells. The new lead candidate, CDK-TCIP2, exhibited sub-nanomolar killing EC50 value and a metabolic half-life in mice of around 3 hours, And while this is of course not sufficient for an actual drug, it was permissive for in vivo experiments, especially given its high potency. More importantly though, we were encouraged by the fact that we can dramatically improve the PK properties of our TCIPs using targeted medicinal chemistry. Now to explain what in vivo model we chose, I have to take a quick step back. In normal physiology, BCL6 is the master regulator of the germinal center or GC reaction that is required for T-cell dependent antibody affinity maturation in the spleen. And similarly to DLBCL, germinal center B cells overexpress BCL6 to repress apoptosis, and germinal centers thus represent the healthy counterpart to cancers with deregulated BCL6. And given the BCL6 targeted specific effects on transcription and apoptosis, we hypothesized that CDK-TCIP2 would specifically suppress the proliferation of GCB cells in a mouse immunization model. And gratifyingly, flow cytometry of harvested mouse spleen revealed that CDK-TCIP2 ablated GCB cell formation in a dose-dependent manner, the best results being achieved using 2 daily doses of 5 mg per kilogram IP, likely owing to the short metabolic half-life of the drug. Importantly, in contrast to other B-cell targeting therapeutics that generally abolish all B cells, CDK-TCIP2 has no significant effects on naive and memory B-cell populations, which agrees with the high specificity we found in the PRISM study. In line with this, we observed no adverse effects or body weight loss in the mice over the course of the 9-day dosing regime, which is notable given the high general toxicity of other CDK9 targeting drugs. The potency and specificity of CDK-TCIPs thus translates from cells to mice, and we are excited to further explore these compounds in models of inflammation as well as of lymphoma and other cancers.
Thanks, Roman. I want to take you through the mechanism of action in more detail inside living cells. To understand this, we first assayed gene expression changes. As we hypothesized, CDK-TCIP dose-dependently increased the expression of BCL6 regulated pro-apoptotic programs. These are highlighted in red here on the right. You will notice that the molecules caused many different overlapping BCL6 target programs to increase. This is, in short, a programmatic activation of multiple pro-apoptotic genes. That feature might be important in later applications of the molecules to mitigate the emergence of resistance in cancer cells treated with CDK-TCIP. You could imagine that the cell must now circumvent many different apoptotic genes being induced, not just one or two, to resist the therapy. We're exploring that mechanism a little bit now further. But next we use chromatin immunoprecipitation sequencing, or ChIP-seq, to quantify what occurs to CDK9 on DNA inside a living cell. This technique uses an antibody to immunoprecipitate the protein and sequence the DNA that's bound with the protein, in this case plus or minus addition of the compound to cells. These experiments first demonstrated that CDK-TCIP addition to cells rapidly relocalized CDK9 to genes with BCL6 summits at their promoter, and the more BCL6 that was bound on DNA, the more CDK9 was recruited. This data shown on the slide is after 2 hours of compound addition at 30 nanomolar. And these loci, these summits, were at those BCL6 target death genes that I mentioned earlier in the presentation, indicating that the BCL6 regulated gene expression pathway, as we saw in the previous slide, was turned on in a programmatic manner due to the localization of CDK9 to BCL6 sites. Now, what was interesting is that at these locations, further ChIP-seq experiments demonstrated that RNA polymerase II was phosphorylated at its serine 2 position on its C-terminal domain. This is the canonical target of CDK9, and biochemical data that I haven't shown on this slide using probe displacement assays inside cells indicated that these effects were occurring at fractional substoichiometric occupancies of CDK9 and BCL6 inside the cell. And so what was this telling us? This was telling us that the bivalent molecule synthesized, as Roman mentioned earlier, from an ATP competitive but reversible inhibitor of the catalytic site of CDK9 was not just relocalizing the kinase to BCL6, as we just saw, but also recruiting its activity to BCL6 bound DNA and its proximal polymerase. We went back to the literature and realized that in the cell, ADP release from kinases occurs about as quickly as our molecule has an off rate for the ternary complex. And of course, as we know, ATP concentrations are extremely high, with some estimates in the millimolar range inside the nucleus of a cell. So we hypothesized that the reversible nature of the bivalent molecule of CDK-TCIP allows for a catch and release mechanism of action. The local kinase concentration increases at BCL6 sites due to compound addition, but the off rate allows for active kinase to phosphorylate, to fall off the ternary complex and then phosphorylate nearby polymerase. This is a working model, but one question that it sets up is, of course, if phosphorylation is actually necessary for the transcriptional and gene expression effects that I showed you earlier. We stress tested that by building a synthetic system to disentangle the effect of kinase recruitment, simple localization, from a molecule that binds the kinase at its active site. To do this, we synthesize a distinct molecule called RCS030207 that binds a ligand binding tag FKBP12, now genetically tagged to CDK9 and recruited using the synthetic molecule to BCL6 sites. By doing this, we could use both wild-type CDK9 as well as a mutated version that is catalytically dead. We assayed gene expression in this system using a BCL6 reporter and found that the catalytically dead kinase cannot activate transcription by simple localization to BCL6 sites. But, in contrast, shown here in red, proximity of the wild-type kinase was sufficient. Those results not only confirmed our core thesis that induced proximity of CDK9 to a transcription factor like BCL6 is sufficient to induce gene expression, but also demonstrated that the phosphorylation effect that I showed you on the previous slide is necessary for the transcription of those genes. Let me now hand it back to Roman to wrap up our story here and take you through how we're expanding this unique mechanism of action in pharmacology even further to different kinases and applications.
There are many kinases other than CDK9 that are involved in the regulation of transcription. An introduction, Sai mentioned CDK12 and 13 that play a similar role in elongation, for example. Hence, we aim to investigate the possibility of recruiting kinase activity beyond that of CDK9 to induce cell death. And indeed, we found that a bivalent compound comprising the same BCL6 BTB domain binder and a CDK12 and 13 selective inhibitor induced BCL6 repressed GFP expression, and this compound, CDK-TCIP3, also killed BCL6 overexpressing lymphoma cells more potently than the combined effect of BCL6 and CDK12 and 13 inhibition. We are thus confident in saying that the principle of turning kinase inhibitors into lineage-specific activators of transcription extends beyond CDK9 and may well be more general. In summary, we're able to show that bivalent compounds linking inhibitors of CDK9 and binders of BCL6 can activate apoptotic genes usually repressed by BCL6 in lymphoma. CDK-TCIPs do not work by target-centric inhibition or degradation, but rather through a programmatic activation of an entire signaling pathway. And precise gene modulation with small molecules is rapidly emerging as a new tool in chemical biology and as a potential therapeutic strategy. Apart from today's presentation on hijacking oncogenic kinases, our team at Stanford has previously reported the recruitment of BET proteins to reprogram BCL6. And in new work that is only under review, but you can find these manuscripts on bioRxiv, we're able to harness the activity of lysine acetyltransferases for gene modulation and reactivate the tumor suppressor activity of p53. Protein-protein proximity is a unifying regulatory principle for cell signaling ranging from stimulus transduction at the cell membrane to the regulation of gene expression in the nucleus. Targeted protein degradation is of course the most advanced modality harnessing this principle, but we think it represents only the tip of the iceberg of opportunities for chemically induced proximity. For example, while today our talk focused on inducing programmed cell death in lymphoma, what fascinates me beyond is that chemical inducers of proximity can be used for highly context and lineage-specific gene modulation in genetically unmodified organisms. The questions ahead are thus as obvious as they are challenging. Can we push the boundaries of this technology beyond lymphoma and apoptosis to other diseases and to other silenced therapeutically relevant signaling pathways? With that, on behalf of both of us, I would like to thank Nathaniel Gray, Jerry Crabtree, and Michael Green for their support and mentorship, Basil Karim, our co-lead in this effort, and the entirety of the Gray and Crabtree labs at Stanford for stimulating and helpful discussions. We gratefully acknowledge funding by the NIH and the Swiss National Science Foundation, and we also note that this work forms part of the intellectual basis of a new biotech startup, Shenandoah Therapeutics, that is based in the Bay Area. Thank you also to Synthesis Workshop for hosting us today, and please feel free to email Sai or myself if there are any questions.
Thank you, Dr. Roman and Dr. Sai, for such an interesting talk. If you like this video, then please consider leaving a thumbs up and subscribe to Synthesis Workshop for more such interesting Research Spotlight episodes. Thank you so much for watching, and we'll see you in the next episode.